RFLP stands for Restriction Fragment Length Polymorphism. It is a molecular biology technique used to detect variations in DNA sequences by analyzing the different lengths of DNA fragments produced when a DNA sample is cut, digested, with specific restriction enzymes. The procedure for RFLP is isolation of DNA. The first step is to extract DNA from the biological sample of interest, such as blood, tissue, or saliva. The extracted DNA is then treated with specific restriction enzymes. These enzymes recognize and cut DNA at specific DNA sequences called recognition sites. Each restriction enzyme cleaves the DNA at its recognition site, creating fragments with different lengths. By electrophoresis, the resulting DNA fragments are separated based on their size using a technique called gel electrophoresis. In this process, the DNA fragments are loaded onto a gel, and an electric current is applied. Smaller DNA fragments move through the gel more quickly than larger ones, causing them to separate into distinct bands. Southern blotting, optional. In some cases, the separated DNA fragments are transferred from the gel onto a membrane in a process called southern blotting. Nitrocellulose membrane of southern blot can then be probed with a labeled DNA probe to detect specific sequences of interest. Visualization and analysis of the separated DNA fragments, whether on the gel or membrane, are visualized using techniques such as autoradiography or fluorescent labeling. By comparing the pattern of DNA fragment sizes among different individuals, researchers can identify variations or polymorphisms in the DNA sequence at the restriction enzyme recognition sites. RFLP analysis was one of the first techniques used to study genetic variation in DNA. It was particularly important in early genetic mapping and forensic science. However, RFLP analysis has been largely replaced by more modern and efficient techniques such as polymerase chain reaction, PCR, and DNA sequencing for most applications. These newer methods provide higher sensitivity, faster results, and the ability to analyze specific DNA sequences more precisely. Nevertheless, RFLP analysis still has historical significance in the field of molecular genetics.